And our first session here is going to be kicked off by Professor Frank Loeffler. Dr. Loeffler is the Goodrich Chair of Excellence in Civil Engineering at the University of Tennessee, Knoxville. Professor Loeffler studies relevant biogeochemical processes in soil, sediment, subsurface, and water environments. He combines cultivation techniques with genetic, biochemical, and analytical metanomics and computational methodologies. It is my pleasure to introduce Professor Loeffler. Professor, the floor is yours.
So thank you very much, Dr. Welton, for showing a picture that was taken more than 12 years ago. So I'm now a little older. So we assembled a team here at the University of Tennessee, Knoxville, to explore if natural attenuation of these toxins that were released, specifically vinyl chloride and butyl acrylate, if this can happen in this East Palestine watershed following this train derailment accident. And as I said, this was a collaborative work between the University of Tennessee, Knoxville, my group, and then Microbial Insights, environmental biotechnology firm specialized in monitoring microbial processes in environmental systems. And then also we would like to thank Purdue and especially Andy Welton for helping us out getting started, because he already made connections to the residents in East Palestine, and that became very helpful for us to collect samples from this near the accident site. So this research was funded through a rapid response research grant from NSF, and we are obviously very thankful to the NSF to making this possible. So this is the team. The people highlighted in bold font are people actually shown on the slide. And then I show here on the right side, Dr. Gao Chen. He took the picture, so he's not in it, but he was a key person in my lab conducting these experiments that I will briefly describe. So collaboration between Microbial Insights, the University of Tennessee, Knoxville, and we had a few people from the Inter-American University of Puerto Rico last summer in my laboratory who also worked on this project. And then, of course, Purdue University for helping with the sampling. So Andy Welton already mentioned the accident released vinyl chloride and butyl acrylate. And those are the two toxins I will be focusing on in my short presentation. I highlight down here polyvinyl chloride, PVC pipes. Those were also on some of the cars that burned, and I talk about this very briefly at the end of my presentation. So 3 days after the accident, right, vinyl chloride, butyl acrylate the cars were compromised. It leaked into the soil subsurface. After 3 days, the decision was made to puncture these cars, release the vinyl chloride, butyl acrylate, and set it on fire. This was a decision that was based on assumptions people made at the time. And the consequence was that there was a massive smoke plume that developed following the burn of these chemicals. And this burn was even visible from an aircraft. So this is a commercial aircraft that was flying over that area, and this was published in Newsweek. So a massive smoke plume. And now coming back to the chemicals that were released when the train derailed and some of these cars were compromised, there was vinyl chloride and butyl acrylate entering the environment and the watershed. Now, vinyl chloride is a well-known toxic chemical. It's a proven human carcinogen, and it has a very low maximum contaminant level of 2 parts per billion. And the maximum contaminant level goal is 0. So this is obviously a concern in the environment and exposure to humans. Butyl acrylate is less toxic, but there's also a risk level associated with it. And there's no MCL established by the EPA, but a minimal risk level of 560 parts per billion. So this is the watershed. So here you see the star, the red star is the derailment site. There's a connector stream that connects to Sulfur Run. Sulfur Run discharges into Leslie Run, and then it goes down to the Ohio River. And this is shown in this inset to the right. So all the blue symbols indicate sampling locations where our team went to this watershed and we collected surface water and sediment samples. So these are concentrations of vinyl chloride that were measured in the watershed after the incident. And you can see in red that sometimes the concentrations were substantially higher than the maximum concentration, maximum contaminant level of 2 parts per billion. But you also can see that the distribution of contaminant was heterogeneous. So certain locations very high, other locations like here BDL means below the detection limit. Similar situation for butyl acrylate. Certain locations extremely high concentrations, in other locations concentrations fairly low. So again, it's a heterogeneous environment, and also the sampling obviously has to deal with this heterogeneity. That explains why certain locations are low, others are very high. So what do we know about natural attenuation, specifically microbial degradation of vinyl chloride in environmental systems? So we know a lot about aerobic vinyl chloride degradation. We know about the pathway, we know about the genes and the enzymes involved. The bottom line is, under oxic conditions, vinyl chloride can be detoxified to non-toxic end products. And organisms involved in this are Mycobacterium and several other genera with species that can perform this process. Under anoxic conditions, so in sediments without oxygen, vinyl chloride can be reductively dechlorinated to ethene. Ethene is a non-toxic product, and the organisms that can perform this process, I list their names here: Dihalococoides, Dihalogenemonas. So these are well-known organisms, a well-known process that can detoxify vinyl chloride in the absence of oxygen. So for those genes and organisms that are involved in these processes, we know enough about them that we can develop specific PCR assays, quantitative PCR assays. Basically, we can go to the environment, extract DNA, and quantify genes that are present in these organisms. Okay, so we can, based on the presence and abundance of these genes, we can predict if this pathway or this process can potentially occur in this environment. And so the yellow stars indicate genes that we know of that are specific for this process of interest, so we can quantify these genes and get information about these processes. So we went to this watershed twice, on June 11th, 2023, and in November 2023. We collected sediment, surface, and drinking water well samples, and we did two different things. We used molecular biological tools, quantitative PCR, to monitor genes, known genes that are associated with vinyl chloride degradation. And this work was mostly done by Microbial Insights using a high-throughput quantitative PCR technology that they use to do this type of work. At the University of Tennessee, Knoxville, we performed a microcosm treatability study. Basically used the sediment that we collected and asked the question, is vinyl chloride, is butyl acrylate degraded by native organisms? So briefly, detection of these biomarker genes using quantitative PCR, this quant-array technology. So in sampling campaign 1 in June and sampling campaign 2 in November, the majority of samples, okay, we tested had organisms that are known to degrade vinyl chloride under anoxic conditions. And what I highlight here in the circle on the right, this is downstream of the accident site. Downstream of the accident site, presumably where more vinyl chloride was available to these organisms, we see higher frequency of detection and higher numbers of these organisms in the sediment, indicating that these organisms responded to the contamination and are actually degrading or benefiting from this release of vinyl chloride. You have to know that these organisms, they like vinyl chloride. For them, it's an energy source. So basically, they are more abundant downstream of the accident site because they responded to this event and utilized vinyl chloride as an electron acceptor, thereby detoxifying this compound. We also detected elevated abundances of these biomarkers for aerobic vinyl chloride degradation. So under oxic conditions, in the top layer of the sediment in soil, organisms that harbor these genes involved in aerobic degradation of vinyl chloride. They were also present in these samples in about same abundances between June and November sampling campaigns. Now, an interesting observation we made is that we also detected biomarkers for anaerobic vinyl chloride degradation in well water samples. Now, this is a little bit difficult to understand. I try to make it easy. So in well water samples, vinyl chloride was never detected. These organisms require vinyl chloride to grow. So why are they in drinking water well samples? What is shown here, right? So we see elevated, not elevated abundances, but we could detect these organisms, so Dihalocucoides in red bars, Dihalogenemonas in green bars, in drinking water well samples. We also detected sulfate-reducing bacteria, and I come back to that on the next slide. Our interpretation is that after the accident, the spill of vinyl chloride degrading organisms basically bloomed because they got a lot of substrate that they need to grow on. That vinyl chloride never reached drinking water wells because it was never detected, but the bacteria that grew in the sediment in the subsurface, they were transported with groundwater into these drinking water wells. And therefore, that's the reason why we detected them. So it's not evidence that there's vinyl chloride in the drinking water wells, but it's evidence of a bloom of these bacteria in the subsurface, and now we can detect them in drinking water wells. And this is consistent with residents reporting that the drinking water smell has changed following the accident. Several residents reported a rotten egg smell of their drinking water. That rotten egg smell is caused by sulfate-reducing bacteria that generate H2S. And I told you, we also detected sulfate-reducing bacteria in the drinking water well water. And this is consistent with this observation that residents reported. So it appears that there's a connection between the water that is contaminated and drinking water wells. And this, of course, is an important observation because it emphasizes the need for hydrogeological information so that we can better predict where do the contaminants go following release, in this case, an accidental spill. Unfortunately, this type of information is not available for this East Palestine watershed. So now I come to the microcosm study, the microcosm treatability study. So we tested or asked the question, is vinyl chloride degradable? So we set up microcosms under oxic and anoxic conditions and then monitored activity. And so what I show you here is evidence for aerobic degradation of vinyl chloride. So the first graph shows vinyl chloride. There's an initial disappearance because of sorption, then there's a lag phase, and then it disappears. We respike more vinyl chloride, it disappears again at higher rate, and so on. So it's quickly degraded. In controls, autoclaved controls, we do not see vinyl chloride disappearance. So it's a biological process. And this happened in every sediment sample we tested. The point is that aerobic VC degradation occurred in all sediment microcosms, so it appears to be a distributed activity. The lag phase is relatively short compared to samples we would take from environments that had no prior exposure to vinyl chloride. In all of these, we detected these biomarker genes for vinyl chloride degradation. And we also noted that this activity is mostly attributed to Mycobacterium species, and those are well-known vinyl chloride degraders under oxic conditions. Under anoxic conditions, we set up microcosms. You can see vinyl chloride disappears. It's converted to ethene. And when we respike vinyl chloride, it's degraded or converted to ethene at higher rates. And see, we see this activity particularly pronounced immediately downstream of the accident site, indicated by this red circle. And when we do some more sophisticated tools, we applied 16S rRNA gene amplicon sequencing, you see that the relative abundance of these organisms, the helicocoides that can degrade vinyl chloride, is increasing over time, indicating that these organisms were enriched under these conditions with vinyl chloride. So we also looked for butyl acrylate degradation, and to make this very short, butyl acrylate is easily degradable under oxic conditions, so this was known. Its fate under anoxic conditions was unclear, so this is what we tested. And in all sediment samples we tested, okay, indicated by these blue symbols, butyl acrylate is relatively fast converted to acrylate and N-butanol, and both of these compounds are further degraded to products that are non-toxic. So the point here is that all of the microcosm studies suggest that butyl acrylate is rapidly degraded under anoxic conditions to innocuous products. My take-home messages from the work that we have done: there's strong natural attenuation potential for vinyl chloride and butyl acrylate in this East Palestine watershed. And this activity obviously should be considered in response actions. So basically the point is that the contaminants released will not last for a long time because microbes can degrade them and convert them to non-toxic products. I also showed you that these molecular biological tools, quantitative PCR, are powerful tools for assessment and monitoring. They provide useful information for decision making. Now, in order to implement these qPCR tools, you must know biomarkers for this process of interest. What this means, we must continue investigations how contaminants are degraded in environmental systems. With that knowledge, we can identify target genes and we can implement these molecular biological tools. If we don't know about the pathway in the genes, we can't do it. The NSF rapid funding mechanism was excellent. It was fast, but maybe it could be even faster. Our first sampling campaign was in June, the accident was in February. We would have liked to sample earlier because we would have gotten a better picture of the dynamic response of the microbiology. Access to site and access to site information was challenging. And I can see, easily how improvements could be implemented. What I mean specifically, we were not allowed to access the accident site. It was fenced off. We had no chance of retrieving samples from the accident site. Accident response guidance documents are currently lacking. I think what I mean is that we have a vinyl chloride spill and there's no protocol for first responders to follow, and that's an issue. We also spoke to many residents, and we noted that community outreach and communication was not ideal, but it's clearly critical because those are the people impacted by accidents like this. This is an image taken after the controlled burn. And now I come back to what I showed on one of my first slides. Vinyl chloride was burned and PVC, polyvinyl chloride. And it's documented that burning of these chemicals or this plastic polyvinyl chloride generates compounds like what I show here, polychlorinated dibenzodioxins. Those are generated in the combustion process, okay? And those chemicals are toxic. They are known endocrine disruptors. Some of them are very strong human carcinogens. They are recalcitrant. They bioaccumulate and they biomagnify. My point is that we had a problem initially with vinyl chloride and butyl acrylate that was manageable. And then what the controlled burn did is generate a problem that is much more difficult to manage and deal with in the long run. So what's needed clearly is a monitoring program for polychlorinated dibenzodioxins and polychlorinated dibenzofuranes, and that probably has to last for decades. We also, our team here, Microbial Insights, University of Tennessee, Knoxville, we work on a fact sheet. This is a draft that summarizes the important information, is easily to understand, and we would like to distribute this to the impacted community and others who are interested in this. This work was published recently in ESNT, and it's a freely available, it's an editor's choice article because of strong public interest, so it's freely available to anybody. So if you want to read more details about what I just talked about, please go there. This is the end of my talk. Thank you very much.